lastly from Jerry Lynn, who will speak on inducing hair follicle organogenesis with defined protein factors. So I'd like to remind you that no photography or video recording is allowed in this session. I really appreciate your cooperation with that. Thank you. Okay, so I'm going to speak to you on the role of beta-catenin signaling in self-renewing stem cells in different epithelial tissues, focusing especially on the hair follicle, of course. So, as you all know, epithelial organs are including hair follicles, the epidermis and sweat gland ducts, and epithelia of the tongue, such as the taste buds and the filiform papillae, are renewed during adult life. And we believe that this ability to regenerate relies on the existence of stem cells that are able both to self-renew and to give rise to progeny who can differentiate. And we're very interested in understanding the molecular mechanisms that control the activity of adult epithelial stem cells, because these go awry in hair loss diseases and other diseases such as skin cancers. So some clues to understanding one of these mechanisms actually came from a number of studies in humans in which it was found that mutation of the human WNT gene is associated with a number of defects that have a regenerative nature. So these patients can show adult onset of thinning hair, adult onset of a smooth tongue appearance and sweating defects. They also often fail to develop a secondary dentition, and they develop palmoplantar keratoderma, illustrated here. Interestingly, a variant of WNT has also been associated in GWAS studies with male pattern baldness. This picture here shows a little girl who's now an adult and is a patient in a dermatology clinic at Penn. And she is a homozygous for a predicted loss of function mutation in the WNT gene. And you can see the abnormally shaped and widely spaced teeth. And her younger brother was even more severely affected. As an adult, this is an X-ray of her jaws, and you can see that she's retained her primary dentition. She also has missing and abnormally shaped teeth and also low bone density in her jaws. She has quite severe palmoplantar keratoderma, particularly on the soles of her feet. And she also has severe nail defects on her toes. So to begin to understand the roles of WNT, we looked at its expression using in situ hybridization in adult epithelia of the mouse. And here you can see in these pictures here, the signal appears as red grains. And if you look at telogen resting hair follicles, there's no signal. But as the hair enters anagen, you can see signal appearing both in the secondary hair germ and in the dermal papilla. And then in full anagen, we see very high expression of WNT. In these non-radioactive in situs where the signal appears purple, you can see that WNT is highly expressed in plantar epidermis and sweat gland ducts, as well as in fungiform and filiform papillae of the tongue. So obviously it's quite hard to study the signaling and mechanism of WNT in humans. So in order to do this, we generated a series of loss of function mutants in the mouse. And these included global loss of WNT, a system in which we can delete WNT specifically in epithelial cells using a keratin 14 promoter-driven pre, and inducible deletion of WNT in epithelial cells using a combination of the first tetrax activator driven by a keratin 5 promoter and a TET-responsive Cre transgene together with this floxed allele of Wnt10A. So this allows us to inducibly delete Wnt10A in epithelia. So what we were happy to see was that these Wnt10A mice nicely mimicked the human patients. Though they had clear defects in hair growth, they also developed abnormal teeth, and teeth were lost after birth due to defects in their roots, but they were also smaller than normal with flattened cusps, and this is also seen in humans. And in addition, the mice developed this smooth tongue phenotype seen here in scanning EM that's also been described in human patients. And about 20% of the mice showed abnormal nail growth as well. So in order to study the mechanisms of action of Wnt10A in more detail, we used our inducible Wnt10A mouse model to ask whether Wnt10A is required for antigen onset. So in this experiment, we deleted Wnt10A in telogen at postnatal day 18, and then we examined the mice 2 days and 8 days later. So you can see that by postnatal day 26, the control mice have started to regrow their hair, as you can see by the dark pigmentation, but the Wnt10A mutants have failed to do so. And if we look histologically, we can see that unlike in the control where the hair follicles are growing normally, the Wnt10A mutant hair follicles have failed to enter antigen. And by Ki67 staining, we can see that they have failed to proliferate normally. The hair actually does eventually start to grow, but it grows in very thin, and it becomes increasingly abnormal with age and is eventually, in fact, lost. At this intermediate stage, you can see the loss of some of the hair and also its greasy and matted appearance. And the reasons for this become clear when we look at the histology. You can see that the epithelial portion of the hair follicle in the mutants has shrunk, but the sebaceous glands, which are stained in this picture as green with FABP5 antibody, are extremely enlarged. And this was interesting because it reminded us of the appearance of hair follicles in human male pattern baldness, where you also see shrinkage of the epithelial portion of the follicle and enlargement of the sebaceous glands. Despite the defects in the epithelium, however, these abnormal follicles retained their stem cells, and we can assay for these using CD34 in green, which you can see is expressed in the control hair follicles and in the mutants. And interestingly, George Kotsarellis' group has shown that a bald scalp in men with androgenetic alopecia also retains hair follicle stem cells. So given the GWAS studies that showed an association of Wnt10A variant with male pattern baldness, we think that Wnt10A defects in Wnt10A may play a role in this disease. So because of the smooth tongue phenotype, we were interested to see what happens to the taste bud cells in the mutants. And differentiated taste buds fall into 3 classes with different functions. So there are type 1, type 2, and type 3, and these can be identified by specific markers. And what we found was that in mice in which we had inducibly deleted epithelial Wnt10A, all 3 of these markers were expressed at lower levels, indicating that Wnt10A is necessary to maintain all 3 types of differentiated taste bud cells. The human patients have also been reported to have defects in sweating. And in mice, unlike in humans, the sweat glands are actually confined to the foot pads of the mice. However, and they have a role more in grip rather than in thermoregulation. However, their general structure is very similar to that of human equine sweat glands. To examine the sweat glands in the sweat ducts in the mutant mice, we separated the epidermis from the dermis and then used Nile blue staining to reveal the ducts. And in this experiment here, we've actually used inducible expression of a Wnt beta-catenin pathway inhibitor, DKK1, and you can see that this causes failure of maintenance of these ducts. And we see the same phenotype if we delete Wnt10A in adult life. So these are the normal sweat gland ducts, and you can see that they're kind of shrunken in the mutant. And this results in a functional loss of the functional ability to sweat. So if we look at the mice expressing the DKK1 Wnt inhibitor compared with controls, here using a starch iodine test, which marks the sweat with this blue, this sort of purple-brown stain, you can see that there's almost no sweating when you inhibit Wnt signaling generally. And in the Wnt10A mutants, we also see much decreased sweating from in the foot pad areas. So Wnt actually can signal through several different pathways. I mentioned the Wnt beta-catenin pathway because this one is the best studied and the one known to have very important roles in appendage development and maintenance. In this pathway, Wnt bind to their receptors called frizzles and to a co-receptor LRP, and this serves to inactivate a complex of proteins that normally targets cytoplasmic beta-catenin for degradation, and thus allowing beta-catenin to accumulate in the cytoplasm and move into the nucleus. And there it partners with left TCF transcription factors, and then this complex activates target gene expression. So the similarity in the phenotypes that we see with Wnt10A deletion or inhibition of the Wnt beta-catenin pathway suggests that Wnt10A is signaling through this pathway. But to test this more directly, we used a Wnt reporter gene in which multimerized TCF left sites are placed upstream of a minimal promoter and GFP. And we crossed this into the mouse mutants. So in this control, you can see that there's nice Wnt activity in the growing hair follicle, but in the Wnt10A mutant, the activity in the follicle is much reduced, even though in the muscle layer here it's still normal, and the muscle does not appear to be affected by Wnt10A deletion. Similarly, if we look at the taste buds, we see decreased signaling in the differentiated taste bud cells. Many of these phenotypes appear to be the result of decreased proliferation of epithelial progenitor cells, and we can see this by Ki67 staining of plantar epidermis, sweat gland ducts, and filiform papillae of the tongue, which all show statistically significant decreases in proliferation in the mutants. And similarly, if we look at the progenitor cells for the taste buds, which are these basal cells surrounding the taste bud, we see that these are also proliferating at a lower rate in the mutant than in the control. So these data tell us that Wnt10A beta-catenin signaling drives progenitor cell proliferation in multiple adult epithelia. And so we've known for a long time that at least for some of these epithelia that beta-catenin signaling is important, but this is the first time that we've really been able to definitively identify a ligand that's important for this activity. So these data drove us to ask whether beta-catenin activity marks self-renewing epithelial stem cells. And to address this question, we used an allele of axin-2 in which an inducible form of Cre has been knocked in. And we combined this with Cre reporter genes. And in general, these are driven by a ubiquitous promoter, ROSA26. And this is upstream of a stop sequence that's surrounded by LOX-P sites that are targets for Cre recombinase, and then a fluorescent reporter. So if we induce Cre expression, this will allow the expression of YFP in any cells which are active for axin 2. And axin 2 is a ubiquitous direct Wnt target gene, so this gives us a readout for cells that are Wnt active. If we initially mark stem cells, then we would expect that if we come back and look at the tissue after many cycles of tissue renewal, then these stem cells and all their progeny would still be labeled. However, if the signaling is taking place in transit amplifying or differentiating cells, then we would expect this label to eventually be lost. So in doing these experiments, it's very important to bear in mind that epithelial regeneration times vary greatly, from approximately 3 days in fully formed papillae of the tongue to up to 48 days in the taste buds. So here we're looking at fully formed papillae of the tongue and plantar epidermis, and what you can see is these clones of GFP-marked cells that originate in the basal layer and are maintained over many cycles of tissue renewal, indicating that Wnt-activated cells self-renew and contribute to differentiating populations in these tissues. In order to look at the hair follicle, we first looked at to see where we would see Wnt-activated cells with our system by assaying 48 hours after initiating induction. And what we can see, consistent with previous data, is that there are Wnt-activated cells in the keratin 15 positive bulge region and in the secondary hair germ, and also in the dermal papilla. If we then look at these follicles that have been induced at anagen onset and examine them at mid-anagen, we find that some lineage trace cells are retained in the bulge. And in addition, we see labeled cells in the matrix, in the dermal papilla, and in differentiating clones of cells in the inner root sheath and in the hair shaft, indicating that the beta-catenin activated cells ultimately give rise to all of these tissues. If we then wait until telogen phase, we can see, and this is now after several cycles of hair growth, you can see that the descendants of Wnt-activated cells are present in the bulge, the secondary hair germ, and the DP. And if we look after in anagen after several telogens, we can again find that this progeny of labeled cells are still giving rise to matrix and hair shaft, as well as other differentiated lineages. Interestingly, we didn't see any clones of labeled cells from the bulge that ended up in the sebaceous gland. Instead, we saw clones of cells that originated in the junctional zone that seemed to be continuous with the sebaceous gland. And by using confetti, which enables more precise clonal analysis, we could in fact identify clones of cells arising in the junctional zone and giving rise to the sebaceous gland. So these data indicate that Wnt-active cells are present in the bulge, the junctional zone, the secondary hair germ, and the DP at anagen onset, and that the progeny of these cells persist or self-renew in all of these areas and give rise to differentiated populations. And we can also say that the sebaceous gland is renewed from Wnt-labeled cells in the junctional zone. We also found self-renewing cells in the sweat gland duct, both in the basal layer and in the suprabasal layers. And when we looked at the taste buds, which, as I mentioned, are renewed from basal cells, we were able to identify labeled basal cells after many cycles of taste bud renewal, and to see that these also give rise to differentiated cells that are not capable of renewing themselves. Because of the nail defects, we also took a look at lineage tracing in the nail, and in the short term we could identify Wnt active cells in the nail matrix, shown here, originating in the basal layer. And after about 180 days after initial labeling, we can see these nice clones of cells that arise in the matrix and give rise to the nail plate and the nail bed. To summarize, WNT controls progenitor cell proliferation in a wide range of epithelial tissues, and Wnt beta-catenin signaling marks self-renewing stem cells in diverse adult epithelia. Because WNT seems to be signaling through the beta-catenin pathway, we think that activating this pathway, for instance, using small molecule activators, is a possible therapeutic approach for some of the defects that we see in the WNT patients. I'd like to thank the members of my lab who did this work, in particular Mingam Zhu, who's a very talented postdoc who did spearheaded most of these studies, and he was helped by Jeremy Harrell, who's a very talented tech. I'd also like to thank our many other members of our lab, our many collaborators at Penn, including our clinical collaborator, Emily Chu, as well as collaborators at other institutions, and our funding support, very importantly. Thank you very much, and I'm happy to take questions.
Please, questions?
Sarah, so do you think that the Wnt-activated cells that you're seeing contributing long-term to the different lineages, are those cells highly proliferative, slowly proliferative? Have you looked at how they proliferate and what their rates are?
So we haven't precisely examined that. We have looked at the turnover rates of the different tissues using the TTA system, and so that enabled us to make sure that when we were assaying, we had definitely allowed the tissues to go through several cycles of renewal, by which those labels would have been lost. So other than the bulge, where we know that there are label-retaining cells, we didn't see label-retaining cells in any of the other tissues. Yeah.
Beautiful data. Sarah, how do these progenitor cells for the sebaceous gland that you've identified in the junctional zone compare to those that Katrin Niemann has described in her previous studies? Are they identical? Is it a different population?
We think they're probably the same. They're positive for LRIG1, so they're almost certainly the same, and our data are completely consistent with her data showing that the sebaceous gland comes from those cells rather than from the
So would you go as far as to say that indeed the hair follicle is in charge of the sebaceous gland, and not the other way around?
It looks like they're emanating from the junctional zone, because we don't see Wnt-activated cells in the short term in the sebaceous gland, but we see the progeny of Wnt-activated cells seeming to
Doesn't really answer my question.
Sorry?
That doesn't really answer
what
my
was your
question.
question?
Who is the boss?
Oh, who's the boss? I think that the cells come from the hair follicle junctional zone.
Yeah, but what does that mean? In terms, who's in control of who?
Well, in this case, not Wnt, because we still see, we in fact see enlarged sebaceous glands.
Thank you. Now you answered my question.
Okay, glad.
So in one of the early slides, you showed that when Wnt is knocked out, that also the fat layer is completely, missing sort of already. What do you think is the interaction there? Is this direct effects maybe from Wnt on the adipocyte itself?
Yeah, that's a good question. So in the slide I showed, the hair follicles were in telogen, so the fat layer is much smaller in that phase. we haven't really carefully looked to see whether there are specific effects on the fat layer. The mice, they have defective teeth, and they're, not entirely, they're smaller than usual, actually. So, there could be some indirect effects on fat, but we haven't looked at WNT in fat, and that would be really interesting.
So, Bruce?
Sarah, I'm over here. I was struck by the fact that you showed the WNT expression in the plantar epidermis. It's like buckets of WNT, right? And then, meanwhile, before we looked at WNT as a placode marker, and you get this local activation of beta-catenin signaling, presumably, to drive the placodes that form the sweat gland. So can you sort of comment on that conundrum?
Because I was so confused about where you're sitting, I missed the first part of the question. But I think you're asking whether there's functional redundancy in embryogenesis, is that?
Well, parts of what I'm asking is if they actually, I'm assuming by their names that they're both beta-catenin signalers.
Yeah, probably.
And one's extremely abundant and everywhere, and yet you get this local response to get placodes. And is WNT not important in that process?
Okay, so what I didn't kind of go through is that apart from the tooth defects, there are essentially no major defects in embryogenesis for these organs. So if WNT is important in embryogenesis, which probably is, it's almost certainly redundant with WNT. So I would think that those two are probably redundant in placode formation.
And is it present in embryogenesis in those,
Is
those high levels, WNT?
Yes. But it's also more broadly expressed than WNT, and that may account for some of the defects in the filiform papilla, for instance, which don't really originate from placodes, and also the palmar-plantar epidermis. So it's required for certain things, but it's very likely to also be redundant for other functions.
Okay,
Okay, thank you.
We have to move on. Thanks very much, Sarah.